When we look at a membrane, we look at a lipid bilayer, two layers of phospholipids that have come together to function as a unit. And it's the tails of those two layers that are facing each other, sort of sequestered away from the water that's found both outside of a cell and the water that's found in the cytoplasm. So we know that all membrane in cells and around cells has this configuration, this lipid bilayer. What I'd like to talk about now is the asymmetry between these two layers of a membrane. It turns out that the anatomical structure of each individual monolayer in a membrane is different. We can talk about the outer monolayer, which is the part of the membrane that's facing the outside of the cell, and the inner monolayer that's facing the inside of the cell. The reason that they're asymmetrical has to do with the distribution of the different types of phospholipids and the different types of proteins. Each monolayer can have different compositions of these things. Now, remember that individual phospholipids in the membrane are not covalently bound to each other. Membranes are very fluid, and the phospholipids have the ability to move around. They're held together through weak interactions. Hydrogen bonds between the heads, van der Waals interactions between the tails, but the individual phospholipids can actually sort of move and travel within a membrane. The asymmetry that we see between the two monolayers, the outer and the inner monolayer, is maintained as asymmetrical because there's a lack of movement between the monolayers. In other words, we'll see phospholipids and proteins sort of bobbing around and moving within each monolayer, but we don't see movement between the monolayers. The kind of movement that occurs within a monolayer gets a name. We call that lateral diffusion. In other words, side-to-side movement within a monolayer. What we don't see is transverse diffusion, or lipids and proteins moving from one monolayer to the other monolayer. Take a look at the image on this slide. We're looking at a little sliver of membrane. You can see that there's a monolayer here and a monolayer here, and together they make up the lipid bilayer of a membrane. The tail region is represented in this light yellow color. The heads have this sort of a peach color. We can imagine that this is the outside of the cell, and this is the inside of the cell. Now, I want you to notice that the outer monolayer has these strange little structures attached in places. These are carbohydrate chains. Remember, we talked about glycolipid, and we're going to find glycolipid in certain types of cells, cells in the nervous tissue, for example. We also find carbohydrate chains attached to the heads of ordinary phospholipids. You can see a whole range of proteins embedded in this membrane. All of these brightly colored structures represent different types of membrane proteins. You can also see these green structures here. This represents cholesterol in this membrane. Now, think about the structure of this outer monolayer compared to the structure of this inner monolayer. And you can see that they're different. You don't see carbohydrate chains in the inner monolayer. They're limited to the outside. And that makes sense because carbohydrate chains tend to be involved in cell identity and cell-to-cell signaling. So it's the outside layer of the membrane that needs these carbohydrates on it, not the inside. You can tell that the population of proteins is a little more varied on the inside monolayer. Some of these peripheral proteins, this one here, they're only present on the inside, not on the outside. And that's probably because these proteins are involved in some sort of internal processes in the cell. They're not needed out here. They're needed inside the cell where these metabolic processes are going on. So we see a clear asymmetry in living cells between the outer and the inner monolayers of membranes. Remember, we currently hold true the idea of the fluid mosaic model of a plasma membrane, and that model was developed by Singer and Nicolson back in the early 1970s. Prior to that time, a couple of different models were proposed for what the membrane looked like. We knew that membranes had a lot of phospholipid in them. We knew that membranes had a lot of protein in them, but we didn't know how those two components of the membrane came together. It wasn't until we had more advanced techniques in the laboratory and more advanced microscopy that we were able to finally get a accurate vision of what the structure of the membrane looked like. And it was Singer and Nicolson who put together this model that we still use today, this fluid mosaic model. Remember, those words, fluid and mosaic, refer to features of the membrane. The fluid term refers to the fact that within a monolayer, the phospholipids are actually moving around. The proteins are actually moving around. They're not staying in the same place. The mosaic part of that name comes from the fact that the proteins don't follow a particularly ordered structure in the membrane. They're sort of placed randomly throughout the membrane, the way a mosaic would be put together in art. One of my favorite analogies of the fluid mosaic model is what's called the pool analogy, and that analogy comes out of a textbook. The analogy has us imagine a pool, like a backyard swimming pool, that we have dumped hundreds and maybe thousands of ping pong balls into, so that the entire surface of the water is covered in ping pong balls. Now, if the water in the pool were moving, that would allow that surface of ping pong balls to sort of flow and ebb and flow on top of the water. Each ping pong ball on top of the swimming pool would be representative of each of the heads on the outside surface of a membrane. Now, if I were to reach into the pool and take out one ping pong ball and let's say color it red with paint, and then place it back into the pool. Let's say I placed it right at this location. If I then walked away and came back an hour later, that ping pong ball would not still be in this spot. Because of the movement of the water underneath the ping pong balls, that red one may have traveled all the way over, say, here by now. each individual ping pong ball is not attached to the other ping pong balls, is it? Each individual ping pong ball can actually bump and move and travel through the other ping pong balls, just like the individual phospholipids in a monolayer can move over time. Now, the other part of the analogy involves the proteins. Remember, the proteins are also sort of floating unattached in the surface of the membrane. Imagine that you took some pool toys, maybe some beach balls or inflatable pool toys, and you threw them in to the mass of ping pongs. Over time, those individual pool toys, they're not going to stay in the same spot either. If you put a beach ball at this location and then you came back an hour later, that same beach ball would have sort of pushed its way through the ping pong balls, and maybe it would be over here an hour later. The same is true of the living membrane. Each of the components, the phospholipids and the proteins, are moving in this membrane, and that's what makes the membrane a fluid mosaic. On this slide, you can see another drawing of a little piece of a membrane. I like this drawing because it's drawn in some of the things that we've been talking about today regarding the variety of structure in a membrane. So as usual, we have two monolayers that have come together to make a bilayer. We can imagine that this is the outside of the cell and this is the inside of the cell. We can also see a whole variety of protein through the membrane. We can see those carbohydrate chains, again, on the outer monolayer, as we're accustomed. There's also a structure in here with the circles. Don't worry about this structure. This represents some of the cytoskeleton. Don't worry about that right now. I just want you to pay attention to what's happening in the different monolayers in this membrane. Take a look at the outer monolayer and notice that the heads are not all the same color. Some of them have this sort of a peach rose color. Some of them have more of a lavender color. They're not all identical. Look over on the inner monolayer. Some of these heads are pink. Some of them are brown. What are we looking at? We're still looking at phospholipids. We're still looking at head regions. Remember, those are just a glycerol and a phosphate and some other small group, remember? So perhaps the rose-colored phospholipids, suppose those are phosphatidylserine, and maybe the purple ones are phosphatidylcholine. Maybe the brown ones are phosphatidylinositol. They're all phospholipids, they're just different varieties of phospholipids. And you can see that there are regions within the monolayer that are different from each other, and there are differences between the monolayers in terms of those types of phospholipids. The proteins are going to be different in the different monolayers. Some of these proteins are peripheral. They're only found on one side of the membrane. Other proteins traverse the entire membrane. But look, sometimes proteins look one way on one side of the membrane. They cross the membrane and then they look differently on the other side. This is the kind of asymmetry we're talking about. There's an asymmetry in the type of phospholipid. There's an asymmetry in the presence or absence of carbohydrate. There's an asymmetry in how the cholesterol is distributed, and the cholesterol is shown in red in this diagram. And there's an asymmetry in how the proteins are distributed. It makes sense because things that are encountering the membrane on the outside of the cell are going to be different than things encountering the membrane on the inside. Processes that are going on out here are going to be different from processes going on in here. It's still a bilayer. It's still a functional bilayer, but structurally speaking, the outer monolayer and the inner monolayer are going to be very different from each other. It's interesting that we still consider the fluid mosaic model to be the correct model of membrane structure, but we have made some updates to it. Unlike what Singer and Nicolson proposed, we now have good evidence that there are regions of membranes that are not as fluid as other regions. So, related to the mosaic nature of membranes, this fits. In other words, different pieces and parts of the membrane, different areas within a monolayer are going to have different fluidity associated with them. Proteins and clusters of different types of phospholipids are going to vary in their fluidity. For example, there are regions that we now call lipid rafts within membranes. And functionally speaking, the phospholipids and the proteins that are within this lipid raft area are going to stay together. So that goes against our pool analogy. What a lipid raft is, in the pool analogy, is a group of ping pong balls and a group of pool toys that hang together over time. So that kind of flies in the face of what Singer and Nicolson proposed. Now, why do lipid rafts stay together? Why do certain parts of the membrane sort of stick together, functionally speaking? Well, we believe it has to do with what that area of the membrane, what its function is, what its job is. So some membrane proteins and some phospholipids are really quite immobile. That's going to allow for things to attach at that location and not change space over time. It's an update to Singer and Nicolson's model. Let's take a look. Lipid rafts are areas of what we would consider a homogeneous makeup within a monolayer. We tend to see them primarily in the outer monolayer. We see clusters of one or two types of protein and clusters of one or two types of phospholipids, and they hang together. Lipid rafts are believed to function as a type of receptor. So structurally, it's important that they stay together, those particular phospholipids, those particular proteins, so they can receive information. Take a look at this drawing, and you can see one of these lipid rafts. Here's that same sliver of membrane we looked at a couple of minutes ago with the yellow tail region, the green cholesterol, and so on. What you're looking at right in the center here is a depiction of a lipid raft. Notice that if you consider the whole membrane, there's a whole range of different proteins. There's this dark teal and this purple and the orange and the pink. But within the lipid raft, there are only two proteins. There's the light pink and the dark pink. And they're clustered. They're several proteins, several individual proteins clustered tightly together. Notice that there's a lot of carbohydrate in that region. Notice that there's an awful lot of cholesterol in that region. Remember, increasing the amount of cholesterol is going to increase the stability of that region, and it's going to help this particular collection of phospholipid and protein stay together. So if it were functioning as a receptor, if it were possible for molecules outside of the cell to come in and bind and interact with this lipid raft, it would be important that all of these individual molecules stay together. Let's switch gears now and start talking about how phospholipids get assembled. How do they get put together in order to build membrane? If you recall from our discussion of organelles, lipids are generally synthesized in the smooth endoplasmic reticulum, or the SER. And the phospholipids are included in that group. There's a very straightforward 4-step sequence to building a phospholipid inside the smooth endoplasmic reticulum, and I've got it outlined here. The first step of the process involves attaching the 2 fatty acids, the 2 tails in other words, to the glycerol. In the second step, the phosphate group is added to the glycerol. Now, that seems like we're done. We're done assembling the phospholipid. But interestingly, the next thing that happens is that newly made phospholipid inserts itself into the membrane of the SER. The importance of that is going to become clear in just a minute. The fourth step of phospholipid synthesis involves the addition of any of those small extra groups that get added onto the head. So if we're going to make it a phosphatidylcholine or a phosphatidylserine, for example, the choline, the serine would be attached at this point. The question is, why are the new phospholipid molecules inserted into the SER membrane? And the answer is so they can be delivered to whatever membrane in whatever part of the cell these new phospholipids are going to live. Newly synthesized phospholipids that are now embedded into some region of the SER membrane will be packaged into a transport vesicle. In other words, that little region of the SER membrane is going to bud off of the SER with the new phospholipids in it. And the transport vesicle will then travel wherever it's going to go in the cell to the place where these new phospholipids are needed. Maybe it's a region of the plasma membrane that needs some new phospholipid, or maybe it's membrane associated with one of the organelles that needs some new phospholipid. The transport vesicle with the new phospholipids in it will arrive at its destination, and it will simply merge in with the existing membrane, delivering the new phospholipids to their new home. Now, that's the way that the phospholipids are sort of replenished in a membrane. The proteins are going to arrive, of course, through a different process because the proteins are coming from the rough endoplasmic reticulum. Any glycolipids that need to be inserted into new membrane are going to come from the Golgi apparatus. Remember, we said that the Golgi is particularly involved in carbohydrate modifications. The Golgi apparatus typically adds or modifies carbohydrate on protein, and it can also add and modify carbohydrate on lipid. So glycolipids, new glycolipids, will be assembled in the Golgi apparatus. And then, again, packaged into transport vesicles to go to their final destination. Take a look at the drawing on this slide. This represents the internal region of a eukaryotic cell. So we're looking into the cytoplasm. We can't see the entire cell in this drawing, only part of it. This represents about half of the nucleus, for example. The purple material represents the chromatin, all dispersed through the nucleus. You can see the nuclear envelope depicted in this green color. Obviously, most of the envelope has been cut away, but you can still see enough of it to see the nuclear pores. Now, attached to the outer part of the envelope is this rough endoplasmic reticulum. Over here, you can see the more tubular, smooth endoplasmic reticulum. And over in another region of the cytoplasm, not attached, is the Golgi apparatus. Now, there's a lot of writing on this diagram. Try to ignore the writing for a minute and just follow the cursor here. What they're showing us is that inside the smooth endoplasmic reticulum, something's being synthesized. And you can see that something as these little purple circles. Remember, the smooth ER is not involved in protein synthesis. It's involved in lipid synthesis. So we can imagine that these little purple balls represent phospholipids. And as they get synthesized inside the SER, when they're finished, a vesicle will bud off of the membrane of the SER with the phospholipids inside. And that transport vesicle will then deliver the phospholipids to whatever location they're going. Maybe they're going to go into the membrane of the Golgi apparatus. Maybe they're going to go into the interior of the Golgi apparatus so they can be modified with carbohydrate. Maybe they're going to be inserted into the plasma membrane. Maybe this transport vesicle will insert them into the membrane of the RER. Remember, all of the membrane in and around a cell is made of phospholipid and protein. So these transport vesicles will deliver the newly made phospholipids anywhere that they need to go within the cell. Let's take a minute and summarize where we are now. We now know how the membrane has structural variability within it. We know that each monolayer, the outer monolayer and the inner monolayer, can be different from each other in terms of what types of phospholipids they contain and what proteins they contain, in terms of how much cholesterol is in that monolayer, in terms of whether or not there are carbohydrate modifications in that monolayer. We also know that the permeability of a membrane is impacted by all those structural features, particularly what's impacting the tail region, as well as one environmental variable, which is temperature. Remember, we said permeability is about whether or not a membrane allows things to get across. And a membrane that has high permeability or more permeability is one that is structured in a way to make it easier for things to get across. A membrane that has low permeability or decreased permeability is a membrane where it's harder for things to get across. We said that how much sterol is in there and how saturated the tails are and how long the tails are, all of those things matter, as does the environmental temperature. All of these things can impact permeability. One other thing that we need to know, though, is this. Permeability of a membrane is not set. The permeability of a membrane is dynamic. In other words, for some membranes, for some organisms, in some domains, Membrane permeability changes depending on the conditions. Now, how in the world could a membrane change its permeability over time? Well, it's because it's regulated. It's regulated. And that means that when certain conditions are occurring around the membrane, enzyme systems can be either increased in their activity or decreased in their activity, and those enzymes can alter the structural features of the membrane. Those enzymes can be, in other words, upregulated or downregulated. They can be increased in activity or decreased in that activity, depending on what's happening around that cell. Things like how hot or how cold it is around that cell. Now, this kind of alteration in membrane structure over time is especially prominent in the types of organisms that don't control their own body temperature. Now, we give a name to these types of organisms. We call them poikilotherms. You and I are not poikilotherms. We are homeotherms. We maintain our body temperature around the same temperature all the time, or within a normal range at least. There are lots of organisms, though, that can't do that. There are lots of organisms that rely on what's happening around them to change their body temperature. Bacteria, for example, fungi, the plants, and even certain animals, including invertebrate animals and cold-blooded animals and seasonal hibernators. All of these types of organisms are considered to be poikilotherms. Their temperature, their cellular or body temperature, is going to be dependent upon what's happening around them in the environment. Now, if you're an organism whose body temperature changes depending on the environmental temperature, you're in a bit of a bind when it comes to the function of your membranes. Let's think for a minute about a snake, a cold-blooded snake. And that snake is quite active during the day when the sun's up and it's warm out. But at nighttime, as the environmental temperature drops, the snake's body temperature also drops and it becomes less active, right? Sometimes in the mornings in New Hampshire, when you go out onto the asphalt roadways, you'll see snakes sort of curled up on the asphalt, and they're trying to draw some of the heat out of the asphalt into their cold bodies. The problem with being a poikilotherm and having your body temperature be impacted by environmental temperature is that your membranes are also going to be impacted by environmental temperature. And that means that their function is going to change depending on whether it's warm or cold outside. Now, that is not a good way to live if you're an organism, if you're a cell, or if you're a multicellular organism. It's not a good way to live because it means that your membranes will function differently during the day and at night as the temperature changes. Now, what you would rather have happen is that your membranes have the same permeability 24 hours a day. But how are you going to do that if you're so impacted by environmental temperature? How are you going to keep your cellular membranes having the same permeability 24 hours a day? The answer is you're going to change the structure of your membranes as the environmental temperature changes so that your membranes can have the same permeability 24 hours a day. This process gets a name. This process is called homeoviscous adaptation. It's the ability for certain cells and certain organisms to alter the composition of the lipids in their membranes in order to maintain the same level of fluidity, the same level of permeability throughout different temperatures. It's a fascinating process. It involves these enzyme systems that can make changes to those three key structures within the tail section of the membrane. Remember, those are the amount of sterol, the amount of cholesterol for animals, the number of double bonds, or the saturation of the tails, and the length of the fatty acids. So let's think of an example. Let's go back to the snake for a minute. The snake's membranes have a certain permeability during the day. A certain, we'll call it a mid-level permeability during the day. The snake is warm because the air temperature is warm. So he's moving around, he's eating, he's doing all the normal things that he does every day, and his membranes are having that certain mid-level of permeability. Now the nighttime comes and the temperatures start to drop. Because he's a poikilotherm, his body temperature also starts to drop. And as we've talked about today, the temperature will also start to affect his membranes. Because he cannot maintain his body temperature 24 hours a day, the cold temperature will start to cause a decrease in permeability of his membranes. And we don't want that to happen. We want his membranes to stay at the same level of permeability 24 hours a day. So what can happen? What can happen to fix this problem? Well, these enzyme systems can turn up when the temperature gets cold, and they can start to make changes to his membranes to bring the permeability back up to that mid-range. Let's think about the things that increase membrane permeability. When it comes to the sterols, like cholesterol, if we're going to want to bring the membrane permeability back up, we're going to need enzymes to start removing cholesterol. When it comes to saturation in the hydrocarbon part of the fatty acids, we said the more the double bonds present, the more permeable the membrane becomes. So we're going to want to have enzymes add double bonds to the tails in the membranes. When it comes to fatty acid length, we said the shorter the length of the fatty acids, the more permeable the membrane. So we're going to want those enzymes to start shortening the length of the fatty acid tails. All three of those things are going to start making the membrane more permeable as the drop in temperature tries to make it less permeable. And that will help hold membrane permeability right in that mid-range where it needs to be.